Secretion of the stomach. The stomach functions primarily as a storage and mixing chamber for ingested food. As food enters the stomach, it's mixed with stomach secretions to become a semi-fluid mixture called chyme or juice. Although some digestion occurs in the stomach, that's not its principal function. Stomach secretion from the gastric glands includes hydrochloric acid, pepsin, mucus, and intrinsic factor. Hydrochloric acid produces a pH of about 2.0 in the stomach. The acid kills microorganisms and activates pepsin from its inactive form, called pepsinogen. Pepsin breaks covalent bonds of proteins to form smaller peptide chains. Pepsin exhibits optimum enzymatic activity at a pH of about 2.0. A thick layer of mucus lubricates the epithelial cells of the stomach wall and protects them from the damaging effect of the acidic chyme and pepsin. Irritation of the stomach mucosa stimulates the secretion of a greater volume of mucus. Intrinsic factor binds with vitamin B12 and makes it more readily absorbed in the small intestine. Approximately 2 liters of gastric secretions, gastric juice, are produced each day. Both nervous and hormonal mechanisms regulate gastric secretion. The neural mechanisms involve the central nervous system, CNS reflexes integrates within the medulla oblongata. Higher brain centers can influence these reflexes. Local reflexes are integrated within the enteric plexus in the wall of the digestive tract and don't involve the CNS. Hormones produced by the stomach and intestine help regulate stomach secretion. Regulation of the stomach secretion can be divided into 3 phases: the cephalic, gastric, and intestinal phases. The cephalic phase can be viewed as the get-started phase, when the stomach secretions are increased in anticipation of incoming food. This is followed by the gastric, go-for-it phase, when most of the stimulation of secretion occurs. Finally, the intestinal phase is the slow-down phase during which stomach secretion decreases. In the cephalic phase, sensation of taste, the smell of food, stimulation of tactile receptors during the process of chewing and swallowing, and pleasant thoughts of food stimulate centers within the medulla oblongata that influence gastric secretions. Action potentials are sent from the medulla oblongata along parasympathetic axons within the vagus nerves to the stomach. Within the stomach wall, the preganglionic neurons stimulate postganglionic neurons in the enteric plexus. The postganglionic neurons stimulate secretory activity in the cell of the stomach mucosa, causing the release of hydrochloric acid, pepsin, mucus, and intrinsic factor. The neurons also stimulate the release of gastrin and histamine from endocrine cells. Gastrin is a hormone that enters the circulation and is carried back to the stomach, where it stimulates additional secretory activity. Histamine is both a paracrine chemical signal that acts locally and a hormone that enters the blood to stimulate gastric gland secretory activity. Histamine is the most potent stimulator of hydrochloric acid secretion. Drugs that block the actions of histamine can lower acid levels. The gastric phase, second phase, is the period during which the greatest volume of gastric secretion occurs. The gastric phase is activated by the presence of food in the stomach. During the gastric phase, the food in the stomach is mixed with gastric secretions. Distension of the stomach stimulates stretch receptors. Action potentials generated by these receptors activate CNS reflexes and local reflexes, resulting in the cascade of events that increases secretion, as in the cephalic phase. Peptides produced by the action of pepsin on proteins stimulate the secretion of gastrin, which in turn stimulates additional hydrochloric acid secretion. And next stage, the intestinal phase of gastric secretion, primarily inhibits gastric secretion. It's controlled by the entrance of acid chyme into the duodenum, which initiates both neural and hormonal mechanisms. When the pH of the chyme entering the duodenum drops to 2.0 or below, the inhibitory influence of the intestinal phase is greatest. The hormone secretin, which inhibits gastric secretions, is released from the duodenum in response to low pH. Fatty acids and peptides in the duodenum initiate the release of the hormone cholecystokinin, which also inhibits gastric secretions. pH less than 2.0 in the duodenum also inhibits CNS stimulation and initiates local reflexes that inhibits gastric secretion. Movement in the stomach. Two types of stomach movement, aid digestion and help move chyme through the digestive tract, mixing waves and peristaltic waves. Both types of movement result from smooth muscle contraction in the stomach wall. The contractions occur about every 20 seconds and proceed from the body of the stomach toward the pyloric sphincter. Relatively weak contractions result in mixing waves which thoroughly mix ingested food with stomach secretions to form chyme. The more fluid part of the chyme is pushed toward the pyloric sphincter, whereas the more solid center moves back toward the body of the stomach. Stronger contractions result in peristaltic waves, which force the chyme toward and through the pyloric sphincter. The pyloric sphincter usually remains closed because of mild tonic contraction. Each peristaltic contraction is sufficiently strong to cause partial relaxation of the pyloric sphincter and to pump a few millimeters of chyme through the pyloric opening and into the duodenum. Increased motility leads to increased emptying. If the stomach empties too fast, the efficiency of digestion and absorption in the small intestine is reduced. However, if the rate of emptying is too slow, the highly acid contents of the stomach may damage the stomach wall. To prevent these two extremes, stomach emptying is regulated. The hormonal and neural mechanisms that stimulate stomach secretions also increase stomach motility, so that the increased secretions are effectively mixed with the stomach contents. The major stimulus of gastric motility and emptying is distension of the stomach wall. Inhibition of gastric motility and emptying is accomplished by the same negative feedback loops associated with the intestinal phase of gastric secretion. In particular, cholecystokinin is a major inhibitor of motility and emptying. Hence, stomach emptying is slower after a fatty meal due to the release of cholecystokinin. In particular, cholecystokinin is a major inhibitor of motility and emptying. Hence, stomach emptying is slower after a fatty meal due to the release of cholecystokinin. In an empty stomach, peristaltic contractions that approach tetanic contractions can occur for about 2 to 3 minutes. The contractions are increased by low blood glucose levels and they are sufficiently strong to create uncomfortable sensation called the hunger pang. Hunger pangs usually begin 12 to 24 hours after the previous meal, in less time for some people. If nothing is ingested, hunger pangs reach their maximum intensity within 3 or 4 days and then become progressively weaker. Alright guys, that's it. Subscribe to my channel please, give me your likes and put your comments on the comment section. Bye bye, see you.